by doing an overview of the Calvin cycle. Let's first start by telling you the Calvin cycle takes place inside of the stroma of the chloroplast. The first part of the Calvin cycle is called the fixation of carbon dioxide. When you fix carbon dioxide, you turn carbon dioxide, which is inorganic, into an organic version of carbon. So let's begin with how this step works. RUBP combines with CO2. In order to do this, it requires the aid of the enzyme called Rubisco. Rubisco helps RUBP combine with CO2, forming our first three-carbon molecule called 3-PGA. We have now fixed carbon into an organic molecule. 3-PGA has 3 carbons, therefore this whole cycle is called the 3C pathway. Step 2 is called the reduction of 3-PGA, and you're turning 3-PGA into G3P, which is down here. To turn 3-PGA into G3P, you need ATP and you need NADPH. NADPH supplies the electrons and the hydrogens in order to reduce the 3-PGA. 3-PGA, when it gets reduced, it eventually becomes G3P. G3P can be used to produce any of the organic molecules you are trying to produce. When this step is done, most of the G3P gets regenerated into RUBP. When this takes place, this requires ATP, and this ATP, in case you forgot, came from the light reactions. This NADPH, in case you forgot, came from the light reactions, and this ATP came from the light reactions. When G3P is converted into RUBP, it requires energy. Now you can start the cycle over again.